let's get started. Welcome all of you back to this second part of this session on the physics of morphogenesis. Continuing in this session will be Professor Erez Braun, who is also at the Technion. And he has over the years been doing some beautiful work on using the ideas of physics to understand the self-organization and the emergence of complexity in a variety of biological settings. And I guess his latest endeavor in this effort is to understand or unravel this process of morphogenesis. And he's going to continue on from where Kinneret left off, I guess. So, Professor Erez Braun, please, the floor is yours.
Thank you very much. So, do you hear me well?
Yes, we hear you well and we see your slides.
Okay, great. So, hello everybody, and thanks for giving me this opportunity to talk about our work. So I'm interested in the phenomena of life, which is not the same as studying biology as a material system. And I hope this will be more clear as we go along. So, sometimes it's worth listening to great poets or writers where they express things that clearly they understand the essence of things better, sometimes better than scientists. So this is a great passage from Thomas Mann, Magic Mountain, and it captures, to my mind, really what is very tricky about trying to understand life, again, as opposed to, in contrast to kind of a material system, by itself very interesting, of course, and fascinating, but life, to understand the phenomena of life, one really needs to go beyond it. As I said, it was something in between the two. It was not matter, not spirit, a phenomena born by matter, like the rainbow above the waterfall, like a flame. So what I want to do, so when I was asked to give this talk, I was hesitating, but then I saw that the title of this conference is really fascinating, because thirsting for theory means that there are thirsty people on the other side, or hungry people, which is very good, especially for students, because thirsty means that there is a gap that needs to be fulfilled. And indeed, what I think is that we are missing a theoretical framework for biology. So I want to, in the first part, which I'll not elaborate too much, but I want to, at least in the form of remarks, give you some idea on the physics aspect of living system due to my perspective and the need for theory. And then the second part, I'll go back to morphogenesis, the subject that Kinneret talked about. Morphogenesis in animal development, because morphogenesis in general has to do also with other forms of pattern formation, and try to elaborate on the nature of the problem. Along the way, I want to emphasize what is really missing and what is the current picture and what is missing in it, again, according to my perspective. And the third part, which I like to save enough time for it, goes back to our experimental effort to try to understand morphogenesis using Hydra regeneration as a model, and using external electric fields to modulate morphogenesis. And to our surprise, we discovered that morphogenesis actually can be reversed. This opens a lot of interesting questions, and this is work in progress, so there will be many more questions that I cannot answer. In general, all this talk, the aim is to raise more questions than answering them. So I think for the students, this is a good opportunity to understand where are the gaps and what we don't understand. And sometimes it's worth more effort to understand what we don't understand than really to understand something. And then I'll summarize at the end. Please stop me if something is not clear and requires clarification along the way. I'll stop in the middle at some arbitrary points according to the spirit of the conference to see whether there are questions. So the need for a theory, this is, I find a fascinating sentence by Eddington, a great scientist, who said in the 30s, long after he made the observation that confirmed Einstein's general relativity, he said, I hope it will not shock experimental physicists too much if I say that we do not accept their observations unless they are confirmed by theory. And this is the reverse of what we usually think of, because usually we think about an experiment to verify a theory. And what Eddington tells us is that if we are lacking a theoretical framework, we really don't understand. And we cannot even interpret the observations in a correct way unless experiment and theory integrate together to give us a complete picture. So a coherent theoretical framework is really essential for our progress in understanding the living world. And I would like to claim without, I think it's true, but we can argue about it. I think that in biology, we don't have a theoretical framework, and we need to work on it. Now, from historical perspective, there was an effort started by Waddington in the 60s to start to develop a theoretical framework. So he organized a series of conferences in Italy, starting in 1966. The end result, along 4 years, the end result was these beautiful 4 books that you see in front of you that are available now. And it's clear from the books that Waddington was really thinking along this line that without a theoretical framework, we are lost in trying to understand life. And I'll come back to Waddington, was a great biologist. Unfortunately, this was all cut off and diminished because of the great progress in molecular biology after the discovery of DNA and all the molecular biology techniques that were developed. And from the 70s, as far as I know, there was no real effort, serious effort to develop theory in biology. So the current picture or the current concepts underlying biological research, which I find interesting to discuss here or to open for discussion, are first that biological systems operate by programs and codes. And in fact, outside of the genetic code, there is no real code discovered in biology anywhere, but people started to talk about codes and programs as a way of thinking about biology, because this was a very useful framework to get progress. And it became more or less the common idea that biological systems operate by codes and programs. The second notion or concept is that reverse engineering of biological system is possible. So we take a hammer, we break a system, whatever level you wish to discuss, organism, tissue, cells, whatever. And the idea is let's make a catalog and find what is the kinetics and dynamics of all the molecular components, and from there build up the theoretical understanding at the end of a biological phenomena. So biological systems are usually regarded as engineered control systems. And all my life in the last, say, 20, 30 years, actually is all my career is devoted to put a question mark on these assumptions. And I'm not the only one on this. Woz is was a famous, it exists a few years ago, was a very famous biologist. He, for example, found the domain of archaea, the RNA world, many contributions in biology. And he writes this because he started to worry himself that if biology becomes a branch of engineering, then we are, if it becomes a branch of engineering, then we are in danger. I think the corona just proved that this is really true. So a crucial difference between natural and engineered control system, there are many differences, but the most important of which is that in a control system, in engineered control system, the way system operates and gets out of errors is by assigning a well-defined target function and then measuring the distance from it and using this distance to feed back on the system and control it. Now, in living systems, there is no a priori target function and no machinery for measuring any distance. So there is no metric that tells the system how far it is from any target. And of course, there is no a priori target also. So when animal develops, there is no signaling or supervisor or anything that tells the system, grow a hand to this size because this is a target and we are so much far from it. And so natural functional systems like biology, like living systems, they must rely on some form of self-organization, which at this stage, I must say, it's a little bit of a cliche because we have to put content in this term. But we have to really think in those directions instead of pre-designed controllers. And the essence I want to emphasize is that we need to close the loop. And to my mind, this is the most important missing link in everything that is, almost everything that is done around in the great progress in understanding subsystems, is that every time you understand a subsystem in biology, you need to rely on another system to stabilize it, to bring material in. So this is a closure problem that is well known in some branches in physics and in mathematics. But I think this closure problem is also a severe problem in biology. And so when we say that the system relies on self-organization, this issue remains empty unless we understand how to close the loop. And I refer to this beautiful paper by Yates on self-organizing systems discussing this. What are possible alternative concepts? I think biology relies a lot on exploratory dynamics, which means that for us, as researchers that want to understand the system, requires shift of focus from structure and kinetics of molecules to closed loop dynamics. And the second thing, alternative concept, is that we need to understand dynamics of systems that are self-referenced, because this is the essence of a living system. It makes reference to itself, and this is something that is very hard to develop a theory towards this, and really is a missing part in our attempt to understand living systems. So along the way, there are two lines of research that I took along the years. One is to work on cell adaptation to something that we call the unforeseen challenge. So it's a concept I'm not going to dwell on here. It's just the idea in a nutshell that if you perturb the system, a living system, by challenges that the system never encountered before in its history, then you discover dynamics that you cannot discover by small perturbation of the system and seeing how it relaxes back to its previous state. And this work is summarized in some papers, but the most maybe elaborated one is this, reviewing reports on progress in physics, the unforeseen challenge. And the second line of research, which I'm going to elaborate on today and complement what Kinneret already told you, is about morphogenesis. Okay. Now, the essence, maybe to illustrate why living system is not just a material system. The earliest reference that I know of, and the beautiful reference, is by Kepler, the same astronomer, who in 1611 wrote this beautiful book, originally in Latin, but you can find it in English. It's a New Year's gift on the six-cornered snowflake. Kepler was worried about finding a present for his mentor who is rich and very clever and has everything. And he asked himself, what kind of present can I bring to somebody that is so clever, rich, and has everything? And then a snowflake fell on his coat, and then another one and another one, all six cornered. They are hexagons, hexagonal structures, sorry, and then he found out that this was the most desirable New Year's gift for the lover of nothing. And the book is about nothing. And this is the essence in biology that is very deep, I think. It's not about the material itself, it's about the organization of the material, and that's what Kepler discovered. So although he couldn't answer the question about the snowflakes, his answer is known to be wrong today. But the book is still beautiful. I really encourage students to read it. But the nothingness that Kepler realized was that the same water molecules, the same material, can be in very different forms with very different properties just because of organization. And this is well known now in physics, of course. But in the context of biology, this is really something that is was trying to grasp. At the end, Kepler wrote that it's also full of humor, this book. So, but I'm getting carried away foolishly in attempting to give a gift of almost nothing. I almost make nothing of it all. So from this almost nothing, I very nearly recreated the entire universe, which contains everything. Okay, so this is Kepler, and the lesson from this is that we have to devote more effort to understand the nothingness, namely the organization rather than the material. Now, the reason for the codes and programs, etc., is not completely accidental. This is just to illustrate the development in biology. By the way, from time to time, give me some signal that you hear me, because
yeah, we hear you.
I have no. I'm looking on a wall here. I don't know. So
no, I understand how that feels. We hear you, and it's very interesting.
there are no cameras open, nothing. So I don't know if I'm talking to myself, which is fine. I like to talk to myself, but okay. So biology and computation actually developed along the way along parallel lines. So I guess most of you know all this history. In biology itself, it started from Avery discovering the DNA is inherited material. Of course, it started much before, but Avery was a key discovery. Watson and Crick on the double helix, Jacob and Monod and others on regulation, and then the genetic code, and then the genome era, boosted in the 2000s by the Human Genome Project and sequencing, et cetera, But in parallel, computation and information branch was developed by major figures like Turing, von Neumann, Shannon on information theory, Wiener on cybernetics. All these guys were very much interested in biology, very much, deeply influenced by them. Von Neumann wrote a book that was published after his death on computer on the brain. Turing, I'll come back to Turing, was very much occupied with a question on biology. Shannon, Wiener cybernetics, concept of regulation is completely inspired by biology. And then there was a gap formed because these guys, along the way of developing computation, thought about complexity. While these guys, or the biology trajectory, took a very reductionist approach, thinking about molecules and genes and forgetting the organization and complexity. But taking these notions of programming and computation and information and codes, etc., biology adapted, or biological research adapted these codes and program as a framework to understand biology. And Schrödinger, the famous Schrödinger from quantum mechanics, had a lot of influence on the development in biology. After publishing this book by him, the famous book, What is Life? The claim, if it can be summarized in one sentence, the claim in Schrödinger is order from order. And what is really fascinating, first of all, Schrödinger was hoping that new laws of physics will come from biology, which as far as I know, till now, there is no new law of physics emerging from biology, but he was hoping that it will. But really amazing, and this is the cleverness of Schrödinger, is that he realized that to stabilize a biological cell and organism, the information must be stored in some form of molecules. And these molecules, to carry information and retain stability, must be some kind of aperiodic crystals. So he envisioned DNA. This is much before the structure of DNA was discovered. It's a beautiful small book to read. Again, I recommend it for all students or not students. It has a lot of clever ideas and shows how by deep thinking, how far one can really get. The problem is that first of all, we don't know any new laws of physics that came up from biology, but also this notion of order from order. I think that if I learn anything from modern research in biology, it teaches me that in fact the problem, the missing link in physics is not order from order, is rather order from disorder. So in a complex system, we know very well in physics how a simple system with a few degrees of freedom can generate complexity. But we much less understand complexity and especially how order emerges from disorder. So order from disorder in physics, which is a major subject in many branches of physics, of course, the boost was understanding thermal equilibrium. So notions like emergent phenomena, symmetry breaking, and order parameters, etc., came up and are more or less well understood by now. Then out of equilibrium systems, what is called dissipative structures, are at the forefront of physics, of course. And sometimes in the 80s, there was some notion that, or feeling that dissipative structure in out of equilibrium system could be understood understood by some very general principles, entropic principles. But then, so for example, out of equilibrium example is fluid flow under two plates with temperature gradient, hot below and cool above. You get convection. So there is symmetry breaking, you can get order, and then you can get turbulence by increasing the perturbation, the external drive. And people saw that this will teach us how patterns emerge in out of equilibrium systems, dissipative system. And I refer to this very beautiful paper by P.W. Anderson and Stein. Anderson is really a very famous physicist in solid state physics that discuss why dissipative structures might be related to life, but they cannot teach us on the theory of life because there is no theory for dissipative structure. Again, in a nutshell, the reason is that all these patterns and structures and broken symmetries, etc., are really controlled by boundary conditions, which certainly is not the case in biology or in living systems. So, so this by itself is certainly not enough as a theoretical framework. And then, as Kinneret already mentioned, more recently, people became very much interested in active matter. These are examples. Kinneret showed this in microtubules. You get active pneumatics, but you can look on birds, coherent flocking of birds, etc. This is a very good, excellent reference. Somebody wants to get into active matter in 2013. So active materials can show self-organized patterns that do not directly reflect the external boundary conditions. So this is a major addition to these passive pattern-forming systems. So we need to take them into account by themselves. However, they are not enough to get us into a theory of biology. So let me summarize the challenges as I see them for the physics of living systems. This is general remarks. First of all, let me say that living systems are historical objects. This is something that we don't encounter in physics. And they are historical objects. I'll come back to it later in morphogenesis, not only because of evolution, because they came from some trajectory in evolution, but every time you observe a biological system, especially if you perturb it, it takes a trajectory that depends very much on the history of the system. And every perturbation you do to it, you really change it. This is, if you want, somewhat analog to quantum mechanics. You do an observation, the system is not unless you do, in vitro, you isolate something, but a living system is not isolated from the perturbation. It can change its trajectory, and it carries with its trajectory some history. And up till now, there is no methodology that I know of that allows to study historical system like that in a systematic way. Now, other challenges is that we lack methodology to study systems with no clear scale separation. In physics, this is one of the major basics that allows to develop a theory. You can separate scales. And I'll talk about it in the context of morphogenesis shortly. So there is crosstalk between levels of organization, which you cannot separate. This is a major obstacle. Another major obstacle is that is the difficulty in identifying what are the relevant variables. What we usually do, we stick to things that we can measure. So we follow techniques. If there is a technique to measure some genes, we look at these genes. And then, of course, declare victory by saying that these genes are the relevant variables. And as Kinneret discussed, we need to go to effective descriptions, and it's not clear what are the relevant variables that allow us to form a framework of a living system. And on top of all this, the complexity that living system exhibit sit at the forefront of physics that by itself doesn't have really a theoretical, a complete theoretical framework. There are hints and progresses along directions. So we don't understand nonlinear many-body dynamics in high-dimensional phase space. I'll explain this in a moment, why high-dimensional phase space is relevant here. We don't understand, and there is no full theory for processes far from thermal equilibrium, and principles of self-organization, especially with self-reference dynamics and closed loops. There is no theory, and there is no theory for that. So let me summarize what the situation in biology. If we want to understand order from disorder, how order emerges from the underlying molecular disorder of the system. So thermal equilibrium is excluded, the system is not in equilibrium. It's possible to think about constraints, but if you think about it for a second, you see that if you increase complexity, the complexity is unfathomable in biological living systems, then the number of constraints you have to add in scales with the complexity of the system. So the number of constraints is huge, and this was a large part of our effort to understand cell adaptation to unforeseen challenge, to show that it's not the constraints of the system that dictates its ability to develop order under unforeseen perturbations. And we are left with dynamic self-organization in active matter, and we have to put content into it. And to my mind, this is if you want to summarize in a sentence, what is missing in biology is to understand the emergence of feedback loops that close feedback processes that close the loop and make a high-dimensional dynamical system collapse into a low-dimensional stable manifold that allows the system to organize itself. And then in low-dimensional systems, there are principles that we understand why the system is organized. Let me just say one word about high dimensionality, because sometimes this is not well understood. So we are very much, we understand very much low dimensional systems, even if they're complex. This is, for example, the famous Lorentz attractor in turbulence, in chaos, sorry. And the reason low dimensional systems are well understood is that if you have an attractor somewhere in a low dimensional system, a trajectory nearby in phase space will directly fall into this attractor. There is no other option for it. In high dimensional system, again, this is not dimension in geometry, in geometrical space. This is dimension in the degrees of freedom of the system, in phase space. So high dimensional system, if there is an attractor somewhere, because there is an infinite number of trajectories that take the system away from this attractor, it's a question of entropy. The system can choose one of these trajectories and will not even notice there is an attractor nearby. And for this reason, people think that high-dimensional systems show slow dynamics, move mostly on cellular trajectory instead of relaxing down, and do not necessarily converge. And this is the basis of beautiful works on glassy systems, etc. So I'm not going to elaborate because this is not a talk in physics, but keep attention that when people use the language of dynamics, they usually adhere immediately to this language of low-dimensional systems, while the system itself, the living system, is not a priori a low-dimensional system. So it goes back to this. We cannot take the low dimensionality a priori. So you cannot take a priori a picture where a small number of degrees of freedom control the system. We need to understand how the system collapsed to this situation. Okay, should I stop here for some question, or there are no questions? People already escape away, or what?
I haven't seen any. Yeah, Vijay has a question. Vijay, please go ahead.
Yeah, so Erez, I have a question about what you said about laws of physics in coming up in biology. This may be a bit vague, so you can correct me. So probably there are no fundamental laws, so to speak. I mean, nothing like quantum mechanics or Newtonian laws. But there are emergent laws at the macroscopic level, at the condensed matter level. And that is something that cannot be derived by just appealing to some reductionist approach. So that would also be as fundamental as the so-called fundamental laws. I mean, they are fundamental at the level that they are being used at. I mean, this may not be well posed, but your comments.
I'm not sure. Your question was cut in the middle, so I'm not sure.
Sorry, I'll repeat that.
Yeah, there are examples in physics for the emergence of order. But again, when this happens, there is no universal complete theory for it. So taking it into the world of the living world, it's not easy. But these pieces that we do understand from physics are very, very important. And so any physical example that can help us to gain insight on new principles of how the high-dimensional system how order emerges from the disorder in high-dimensional systems is very helpful. I don't know if it answers your question or.
No. So maybe you didn't get what I said. So I was trying to ask your comment about Schrodinger's book and no new physical laws being discovered in biology. Okay. So I was just trying to understand when you say physical laws, it probably means something like quantum mechanics or Newtonian mechanics or those fundamental kind of thing. But I was trying to argue that even emergent laws at the level of many body systems, things that come from broken symmetry or emergent phenomena, they're also as fundamental as any other law. I mean, at the level that you're looking at, they're as fundamental as that. This is something
sure.
that remarks.
Yeah, sure. That's why I gave this example.
Yeah.
So I refer to this paper. So Prigogine claimed victory sometime, I don't remember, in the 70s or the 80s, by saying that he understands this emergence of order in non-equilibrium systems. But then these guys analyzed it and tell you that this is his victory is not really a victory because there exists no theory of dissipative structures that do not rely on boundary conditions. Mm-hmm. So, for example, if you have a pattern in fluid flow, if you remove the boundaries, you get turbulence, you get out of order. This is well known. There are many, many beautiful experiments that show this. You need to stabilize the system by either external agents or by boundary conditions. And so it's not an emergent phenomena the way we see it in living systems. Okay.
But the active matter picture, you seem to imply that'll
no. So active systems bring to the table something new that allows the emergence of patterns, stable patterns, without boundary conditions, because you don't have to inject the energy from the, large scale or something. The energy comes from the molecular, from the microscopic constituents. This brings to the table something new. And certainly, a lot of the phenomena that we see in biology are driven by active systems like that. But what I say is that this by itself does not solve the problem. It helps. I mean, it's a framework that we need to hug and work and understand very well. And it's very useful, as Kinneret showed for example, even in morphogenesis. It's very useful. By itself, it does not solve the problem of order from disorder in living systems, to my mind.
Yeah, I agree. Thanks.
Okay.
Thank you. There are a couple more comments, I guess. Sonali, you want to?
Yes, that was very enlightening, Dr. Braun. I have this comment. There are two aspects. One is analysis, breaking off the system into parts, say in this case, life, and understanding its principle. And the other aspect is synthesis, putting the parts together and getting the whole. Now, Feynman, the physicist, as this quote is attributed to him, what I cannot create, I do not understand. So could you comment this in the context of biology? Because we are trying to break something into parts and trying to understand the parts, but we have not yet synthesized the whole yet. So it's like, thank you.
First of all, who am I that I'll comment on Feynman? But I completely disagree with this remark of Feynman, which for me, it's a kind of, a cliche that is completely wrong. We don't build black holes in order to understand black holes. We don't build, you don't have to build a system in order to understand it. This is completely not true. And it's very useful to understand parts of systems if you can take all this beautiful work on in vitro and synthetic biology of course, it's very, very useful, etc. The question, how much it's useful to understand principles that allow us to build a theory, because I want to tell something that is very important at this stage, is that if you build a system that behaves like another phenomena, another natural phenomena, it doesn't mean that the natural phenomena really works by the same principles. You can get to the same result by many different trajectories, especially in complex systems. And by doing this, I have a lot of hot discussions with my friend doing machine learning and artificial intelligence. The fact that they can solve a problem by a computer doesn't mean that nature solves the problem the same way. So I don't have much to add on this. I don't know if it answers the question.
Thank you. Very clear. Thank you.
Maybe you should go a little bit on and then we stop again, because I see that time is running and I really want to go to get to the electric field.
Yeah, let's do that. Let's continue on and then take.
Maybe we should leave some time at the end for questions, because as I promised, there will be more questions than answer. I don't aim to understand it, to explain or give answers to anything new. So let me now jump into morphogenesis, which is the subject that Kinneret talked about, and tell you a little bit about the nature of the problem. So the question in morphogenesis, you start from a fertilized egg, say, and get into a creature, a functional viable animal. There are two questions. There are many questions, but there are two that are completely open at the moment. One is, how does the specific shape of an organism emerge? And the second is, what type of dynamics ensure the robustness of the system? And this again takes me to the essence of our lack of understanding of living system, is a question of how the system closes the loop to ensure, in that case, specific shape and robustness. It's very helpful if one will open his camera, so I can see that I'm not, again, because sometimes I have problem with the Zoom. So patterning in development, there are beautiful work for many years already. This is the embryo of Drosophila, a fly, that people at least understand the mechanisms at the onset of development, with these patterns of different proteins that emerge, that eventually lead to the fly, to the viable fly. And also there is an interesting connection to DNA structures that are connected to this. But all this relies on a few genes that emerge at some order. And by the way, the Drosophila as a model organism is really every model organism is unique in its own way, but the fly, Drosophila, is unique among creatures. And a lot of what we know in development comes from the fly, but actually does not apply to other animals. But there are other examples like the zebrafish. These are now common model systems that people use. The fly was the original, I think, but now zebrafish and C. elegans and mouse and all these become model systems for development. And the idea of the current picture is that there is some a way of breaking the symmetry and the emergence of patterns by chemical molecules that I'll come to it in a moment, and they dictate the rest of development by some hierarchy of processes that end up with a creature that is functional. Now, very early on, actually in the 19th century, people realized that there is a lot of universality in development. What you see here is a bunch of embryos. These are real pictures. This is not an image. These are real pictures of embryos. One of them is human. I guess that it's very hard for you to know which one is human. And when you let them, but you see that they are all have more or less the same structure, more or less the same pattern. And then boom, at some point, they develop very differently. One goes into fish and the other goes into human. So this was the human embryo. Okay, so there is a lot of universality in biology, not only in this type of creatures, the chordates that have a spinal cord, but also in many other developmental systems. And especially one of the breakthrough of the 20th century was the discovery that the molecular processes, the underlying molecular processes are also universal. So they appear that the same processes repeat themselves. And this is, by the way, another recommended book by Gerhardt and Kirchner that discuss this point. You start from very different initial conditions, converge at the mid step to a pattern that looks the same for many, many different organisms, and then boom, there is a divergence. And up till now, there is no good picture, there is no good hint what happens here at this stage that makes this to make a mouse, a bird, a fish, etc. And it was Waddington, the one that I mentioned before. You see that he looks like an intellectual, smoking. Unfortunately, not many people are smoking these days. So it really helps, I guess, to deeply think about problems. He coined the term epigenetic landscape. It was his idea. And he was occupied with this question of canalization, how system converge to a very specific and robust trajectory in development. He envisioned that these genes that you see down here pull on some landscape, and you think about he wanted to think about biology, about development, as a ball running downhill through these valleys that are curved by these genes. Of course, I intuitively object this. It's a beautiful picture, by the way. This book is absolutely a masterpiece by Waddington. But I intuitively object this notion that there is some landscape that is created outside of the process itself that tells the system how to flow, because it all must be the same type of process. This is the notion of self-organization, self-reference, closed-loop dynamics, etc., that we try to develop. The more modern view is that you start with stem cells, and then they branch into very specific type of cells in development. And again, I'll come back to this picture, which was completely shocked and vibrated some years ago. But the notion is that you start from the stem cells and it flows like downhill, one of the Waddington landscapes. From the physics point of view, people adhere also to these pictures, but instead of just a landscape, they took a dynamical system approach, especially these guys. A lot of work recently by Kaneko in recent years, and Kaufman before that look on cell types and the robustness in development as attractors in low-dimensional systems. What bothers me in this picture is that this picture of attractor is really very useful when you think about low-dimensional dynamical system. And I try to convince you that this is not the case for if you want to understand life and development by itself, because you have to think about high-dimensional. So maybe at the end there is a low-dimensional attractor here, but you have to understand how the system converges to this. It's not given. It's not that there is something in the system that locks it to some pattern. The pattern is emerging in high-dimensional spaces. And there are also many open questions about this that I'm not going to discuss here. Let me go very quickly now. So there are two pillars that the common picture of morphogenesis rely on. One is reaction diffusion, and the other is positional information. Let me just briefly mention. Kinneret also briefly mentioned him. Reaction diffusion, the picture of reaction diffusion started by Turing in '52, the same Turing that I mentioned about the computers and computing. He coined the term morphogenes for molecules that lead to patterns. And he said, this is again a beautiful paper, and again, he said specifically, explicitly in the paper, that he thinks only about chemical reactions, and in fact, he knows that this is not enough. But reaction diffusion is about patterning by chemical reactions. You get a bunch of equations for the morphogene concentrations, which have a reaction part and the diffusion part, and the reaction is usually nonlinear. And in more modern picture, one of the elements serves as an activator, and the other is usually an inhibitor. But you can have many different types. And to my surprise, although morphogenes is a driving concept in understanding development, the number of morphogenes that were actually discovered is very, very small. Is a handful, is something two handfuls that are well understood. But the concept is a leading concept in biology. And what Turing showed, but this reaction diffusion, that there are 6 types of stable patterns that can emerge. So with combination of morphogenes, you can get elaborated patterns that are very useful also to understand the emergence of patterns on animal coats, etc. The other concept is positional information, because it's not enough to create pattern of chemical reactions if this cannot be implemented as change in behavior of cells and tissues. And so this was, the idea was developed before Wolpert, but Wolpert in '69 gave a big push to this idea, which is called the French flag model because of this picture. So if you have a source and sink and the gradient, if there is a threshold and in some position in space, this morphogene gradient crosses it, concentration crosses a threshold, then the system transform, and so there is cells that develop into specific type of cells and into tissues. What about physical forces? So as Turing already mentioned, we need to put more in. Kinneret showed you mechanics, aspects of mechanics. This is the guy that, to my mind, started the understanding of physics, the emergence of patterns in physical system by his beautiful and very influential book, Gross and Form. This is Darcy Thompson. This is the original publication. This is the second edition that he wrote, extended edition. And I'm not going, I don't have time, so I don't go into some of these beautiful mathematics that he develops to understand the transformation between organisms. The book was written originally as retaliation to Darwinism, that he rejected the idea that everything should be explained by evolution. He said that physics and mathematics can explain a lot of what we see around. So I'll skip this. So let me just, I'm short of time. Let me do a very quick list showing that morphogenesis actually arises from the interplay between processes at different level of organization. And again, our problem is that there is no cutoff, there is no separation of scales, and there are processes at the molecular level, at the cellular level, at the tissue level, and the whole organism level. And all are connected together, and they span a huge range in scales, both in space and in time. And the reductionist approach is to go into some molecular components here, or say, or some cellular level, whatever level you choose, and try to understand it, which is very useful and very important. But there is an alternative to this, which I think is more appropriate if one wants to develop a theory for morphogenesis, a physical theory for morphogenesis. And this is the effective approach that Kinneret mentioned. You want to coarse-grain processes and try to identify the relevant variables that allow you a coarse-grain or effective description of the system. That's what we do in physics. We always do this. At every level, that's what we do. And let me just put another remark here. For every level here, the level above it serves as the environment for this level. So the cell is the environment for the molecular, tissue is the environment for the cell, et cetera, et cetera. And very important to realize that the environment is an essential part of a living system. It's part of the dynamics. It's not a passive thing. It's not like a boundary condition, because the organism affects the environment. And if you think about environments at different levels, say the cell is an environment for the molecular, the cell is certainly affected by the molecular, so the environment of the molecular level is affected, et cetera, up to the whole organism, which is as the external environment, which is also affected by the organism and feedback on its behavior and dynamics. And this is an example for mechanics. I'm not going to. So this brings me to the last part. I'm not going to stop for questions here. Let me stop after I'll show you the electric field thing. Is that okay? Okay.
Yeah, that's perfectly fine. That's perfectly fine. Yes.
So Kinneret mentioned mechanics as an essential part of morphogenesis. Let me add another ingredient that is essential. I claim that morphogenesis emerges by 3 types of interconnected processes. These are biochemical processes, mechanical processes, and electrical processes, which must evolve simultaneously as coupled field. This is the essence. You cannot take just one of them and claim that you understand morphogenesis. It came up in Kinneret's talk that it's very important to understand the mechanical aspects, say by actin and defects, but then you have to bring biochemical processes in. But you have to bring also electrical. Every cell in an organism is affected by electrical processes, voltage gradients and ionic currents. And there are many beautiful experiments over the years that show that electrical processes are inevitable part of morphogenesis. Okay? This is in wound healing, for example. It becomes now a very important part of physiology, and also as a therapeutic technique to understand the emergence of voltage gradients and currents, electrical currents, as a way to understand wound healing is essential part of the process, so it's not disconnected from morphogenesis. I'm not going to elaborate on Hydra because you heard already. For those that hear it for the first time, it's a multicellular organism, a behaving animal, a predator, and these are the scales of an adult, more or less. It can vary, but this is more or less the scales of an adult animal. Okay, and the guy that started actually all issue of regeneration is this guy, by the way, a mathematician from Geneva, Abraham Tremblay, who was a tutor of these two kids in Hague, in Holland. These are kids of a duke, and they worked in the ponds, in the property. He was not the one that discovered Hydra. Hydra was known before, but he asked the question, what happens if I cut Hydra? And he was the first one to discover regeneration, and he wrote a beautiful 4 books. And this is considered as the birth of experimental biology, because he teaches in these books how to write a lab book, how to do a control experiment, what's the importance of a control experiment. He really started to develop, because up to this point, 1744, biology was an observational field, a natural observational field, and not an experimental endeavor. So this is Hydra, and we are going to concentrate on this part, the epithelium, which wraps the animal, and I'm going to discuss what's going on in Hydra regeneration under external electric field. So this is work in progress that started by an observation by us that electric field perturbs morphogenesis in a way that I didn't see before that, and gives us an opportunity to try and understand the integration. So by using external electric fields, it gives us an opportunity to try to understand these integrated processes. Electric fields in development is not completely new. It started by Raus in the 19th century, beginning of the 20th century. He put electric fields on eggs, on oocytes. He didn't discover anything, so it's not very well published, but he tried. And then this guy, Baker, was a doctor, medical doctor, started to think seriously that we should understand the electric fields, for example, in bone regeneration, and etc. So it's a beautiful thing. This is a curious paper in Science he wrote on Hall effect in salamandra. For physicists here, to find Hall effect in salamandra is really unusual in the 60s, but that's what he did. And more recently, a lot of work by the lab of Michael Levine in Tufts shows that if you perturb electrical processes during regeneration, you can get all kinds of curious things. You can go to his lab site to find out all of a kind interesting experiment that show that if you perturb, in that case, he perturbs the internal. He doesn't use external fields. He uses drug perturbation to perturb some internal electrical processes. Let me just mention, because I'm fascinated by this. This is a company, NovoCure, by the way, an Israeli company, but I'm fascinated not because it's an Israeli company, but because this is the best, as far as I know, the best cancer treatment these days, ever. I mean, really, it takes people with a fatal tumor, brain tumor, and completely relax the tumor without relapse for many years. You take with a suitcase, backpack with the electronics. You have electrodes on your head. It uses AC fields at very high frequencies, not very high, but high frequencies, hundreds of kilohertz. And they show that this stops cell division. Because in the brain, for example, cell division occurs only in tumors, in cancer cells, or mostly, it just stops cell division and kills the cell. And so tumor is more or less a repair. Now, Hydra is a very curious creature for many different aspects, but also its electrical activity is very important, is very peculiar and rich. Let me just say, because lack of time, that almost all type of cells in Hydra are electrically excitable, so they can generate reaction potential, especially the epithelium, which is a kind of muscle, as Kinneret discussed. All epithelium cells can generate action potentials. And once I realized that, and the reason people don't use much external fields is because it's very hard to penetrate and perturb large systems with electric fields because it doesn't penetrate well. Because the epithelium is excitable, it occurred to me that even weak electrical perturbations can be amplified by this machinery, and so even weak penetration of electric field can cause an effect, and there is a lot of work done during the years to show the electrical properties of Hydra. It can generate, as I said, action potential. All the cell can generate. This is a full Hydra. These are action potentials. Hydra is a nerve system, distributed nerve system, but there is a way to grow Hydra without nerves, without neurons, and even Hydra without neurons generate action potential, so it doesn't come from neurons necessarily, although all neurons, of course, can generate also action potentials. And let me show you the phenomena we discovered. I'll run it quickly because I think we are running out of time. So the experimental setup, I'll come to it in a moment. You have two electrodes. This is a spheroid. So it's a small Hydra tissue folded into a spheroid, the way Kinneret explained. This sits in a well curved in a gel, and under external electric field, we use AC. We demonstrated the same phenomena in DC, but this is not very good because the system gets contaminated very quickly. We use AC fields of around kilohertz, and this is the phenomena we found out. Let me run it quickly. So 0 voltage, you have the voltage written here, you have the time on the left. This is what Kinneret showed you. There is Hydra develops, and then we put a field, 20 volts in this case, and the full structure goes back. You can see it here. It goes back into some kind of a spheroid. Then you drop the voltage and it develops again. Then you bring up the voltage again and it goes into a spheroid again. You drop the voltage and it develops again. Once I saw this, I was completely shocked because it means that you can modulate and control morphogenesis to some degree at least by external physical field. And this is not only fascinating, I think it opens many new possibilities to try to understand the system. So let me go very quickly, let's say 5 or 10 minutes over some experiment to show you what we learned by using this technique, trying to understand to what extent we can modulate morphogenesis on demand, and answering the question, is morphogenesis really a reversible process? Okay, and the classical picture tells you that you cross thresholds and symmetry breaking, and because of the complexity, thinking about going back, not necessarily through some specific trajectory, but unfolding is very curious and opens many possibilities. So this is a setup. I'm not going to go into experimental technique. What we have now is a pair of electrodes per sample. So there are many samples in parallel. under a time-lapse microscopy. We have a multiplexer that allows to control, in principle, the voltage and frequency for each sample. Because of the variability and history dependency of each sample, we want to sometimes perturb some tissues, not perturb other tissues. And the system is under continuous medium flow to prevent any contaminations or things like that. And this is the basic phenomena. You can either halt regeneration. So there is a critical field that is frequency dependent. I'll say something about it in a moment. And it's tissue dependent. So it's individual tissue, different tissues will exhibit individual critical fields. And by now we know that the field, it's not the voltage is important, but this is not so important for this talk. And once you approach this critical field in a gentle way, you can halt regeneration. You can hold the system at this stage without letting it regenerated for hundreds of hours. While in a regular experiment, within 48 hours or so, the experiment is over because the system is developed and that's it. And then you can start asking questions about the status of this and what's the meaning of regeneration, what switches on regeneration, what is stored in memory, etc. The other trajectory that is sketched here, and you see a big picture here, is that you let the system develop, you come to the critical field, you cross it, and then you take the system back and let it develop again. And this shows you, for example, a trajectory like that. So this is at high voltage. The system is barely at the edge of development, but it doesn't develop. You drop the voltage to 0, it develops. Then you increase voltage again. Then you can stop it and go back. You drop the voltage back to 0. Shows that the tissue didn't lose its potential to regenerate. We didn't kill the system. We don't destroy the process. We just perturb it. Okay? So I'll skip a lot of things here. One important and interesting step that we are far beyond understanding it, you start with some pattern that emerges by letting the system to regenerate. You take it back and let it start again. And it seems from many experiments that the system starts a trajectory that is not following the previous one. It can develop a different number of tentacles, different body shape, whatever. Okay? So there are things that's stored in memory. We didn't erase everything, but the system has enough flexibility to start a new trajectory. And this is fascinating because this means that you can start from the same tissue, giving its history, different trajectories of development and study the potential of the system to develop rather than just a simple one realization of it. And I'll come back to it in the summary. Okay, and you can repeat this cycle. The record is 5 times, but this is because the system, as Kinneret also showed in regeneration, it loses cells from time to time because it needs energy. There is no energy in the medium. The medium is just very low osmolarity medium, 1 millimolar. There is no energy source from the outside, so the system needs to grab its own cells for the energy needed. But it raises a lot of questions in physics, where the energy comes from, how much energy is needed, et cetera, that you can start to explore. Now, one important thing is that calcium plays an important role in all this. So what you see here is a transgenic animal with a probe, fast probe for calcium. This again in the spheroid case, you see here the time, and you see that it has burst of calcium. So the fluorescent you see is a calcium probe, and we are in the process of trying to understand all these signals. I'll say a word about it in a moment, but you see that you can increase, don't know if you see, if you increase voltage, it started to burst like crazy. So calcium reacts, I'll show you this in a moment, calcium reacts to the external field. What we have shown is that every calcium excitation is driven by a burst of electrical excitations, but the opposite is not true. You can get, every electrical excitation leads to burst of calcium excitation, but the opposite is not true. You can get calcium activity without electrical spikes. What you see here is a burst of action potentials measured directly on the tissue, and in orange here are bursts of calcium spikes that follow. So under certain conditions, or under proper condition of an experimental condition, calcium really faithfully report on the electrical activity of the tissue. Now, there are many reasons to look on calcium. It's a very important signal. It faithfully reports, as I said, on electrical activity. Is a universal effector in many biological systems, and especially for us, calcium is a kind of signal that can serve as a mediator between biochemistry, mechanical, and electrical processes. So it's very important to follow it. And we can relate somehow the shape and the stages in regeneration to the calcium activity by doing fast time-lapse microscopy, fast enough. Okay? So this is summarizing the fact that once we increase the voltage, calcium activity is increased. The two colors here are two different experiments that to show that this is very robust. This idea that calcium reacts by increasing the electrical activity of the tissue, the calcium activity goes up. And you can relate this to morphogenesis and going back. Although we don't understand the role of calcium in this process, but empirically you can map calcium activity to stages in the behavior of the system when we modulate it. Although again, we are still lacking even minimal understanding what calcium is really doing. This is summarizing many experiments. It shows that the calcium indeed under voltage external field, the calcium activity goes up by much. This is a logarithmic scale, and this is distribution of the fluorescent density of many tissues under either zero voltage in blue and high voltage in red. There is a frequency cutoff. I'm going to cut this short. We have shown that around 3 kilohertz, the system becomes transparent. You can see it here, but you can see it also, the system starts to develop again. If you go to high voltage and take the frequency to above around 3 kilohertz or around 3 kilohertz, this is very different regime from the Novocure I showed you before that treats cancer. We don't have to go to high frequencies. It's a very different phenomena. And I can elaborate a little bit on it if you ask questions. This is again summarizing many experiments. It shows that if you go zero voltage, you get this distribution of calcium activity. If you go to high frequency, you get a zero voltage. The system becomes transparent to the external electric field. And this is in mid frequency, it becomes very sensitive to the external electric field. And again, because of lack of time, I guess I need to wrap up. The problem of calcium is very complicated and very interesting because the calcium has a spatiotemporal dynamical patterns. It's not that the whole tissue behaves as a whole. What I showed you before was an average signal over the entire tissue. We can go now and measure the spatial patterns that emerge. And what you see here, for example, is that the standard deviation that you show you in red, the standard deviation of the signal across the tissue does not behave exactly as the mean, although every time there is a spike in the mean, of course, there is a spike also in the standard deviation, but this is logarithmic. Look on the relaxation here and the way it behaves towards the next spike. So starting to analyze the spatial temporal dynamics can teach us a lot about the system. And because this is going to be, I guess, on the recorded or whatever, you can go back and look on the slides and look what we can measure by now, even using very, very shallow statistics, just the second moment of the distribution. And there are also interesting correlations. This is a temporal correlation that shows, so this is a temporal autocorrelation. When you increase voltage, it starts to oscillate. You see it in the beautiful oscillations in this correlation function here. And there are very interesting also spatial correlation that you can measure that I don't have time to get into much. Let me just say, which I forgot to say, is that there is a curious adaptation that occurs. And the curious adaptation, you can see it also on the mean, but also is the fact that if you go on cycles, so you reverse regeneration, and then you want to let it go again, and then reverse it again, you have to keep increasing the critical voltage, the critical field that causes reversal of regeneration goes up and up and up every time you do the cycle. So the tissue adapts to the previous conditions, and we can see this adaptation very clearly also on the spatial temporal dynamics. I'm not going to get into it, but this adaptation is a major important issue, as I said, because it's an historical object. Its exposure to previous perturbation is somehow imprinted, instilled into the system, and causes changes in its behavior in future trajectories. Now the question is, can I have another 2 or 3 minutes to finish this, and then?
Yeah, sure.
Because
sure,
I already, I think, more or less. So,
no, please go ahead. Please finish your slides. Yeah.
Okay. So one important question was whether reversal of morphogenesis is just in the morphology, or there are signatures that we can observe in the underlying signaling, say, biology. So let me just in half a minute tell you about the herd organizer. Kinneret mentioned it. So Ethel Braun, doing work on Hydra, in fact, found out in the beginning of the 20th century that some parts of it can induce, or a part of the head can induce an extra axis in Hydra. And Spemann and Mangold was a PhD student of Spemann. They used tadpoles and other animals, later in Salamandra, to show that indeed there is a group of cells in developing animal that serves as an organizer. It's a very important concept in development. The organizer is an inducer. He got the Nobel. So there are 2 women and 1 man. So you can guess who got the Nobel Prize, who are completely ignored. So Spemann got the Nobel Prize for it. Ethel Mangold did a lot of the work as a PhD student, but the whole idea started from Ethel Braun, which they never bothered to mention even, but the whole idea of induction comes from this woman. And the organizer is very important, and especially in Hydra, because Hydra actually throws cells all the time and refresh its entire tissue structure, it needs an anchor. So it will not change its body shape every time it does this. And the anchor is the head organizer at the tip of the head, which again, the organizer appears in any almost any developmental system. But in Hydra, it gave an opportunity to study a little bit of the mechanism, or molecular mechanism. So the head organizer in the tip of the head that is marked here by these signals allows Hydra by induction to actually for cells that are motile along Hydra or change and need to replace another parts of tissues, it gives them an orientation and polarity information about the axis, the body plane, the body axis of the animal. And people made a lot of effort to understand the mechanism. And one of the most important players is a process called Win3. It's a gene that is part of the organizer, and it's autocatalytic. And people have shown that Win3 is a major part of the organizer. It's not the only part, there are many other parts, but it is the one that regulates the system by autocatalytic kinetics, stabilizes the organizer. And what we discover, again, to make it short, so there is a transgenic animal with a marker for the Win3. Unfortunately, it does not mark Win3 itself, it marks the expression activity of Win3. And it has a lot of problems because it has delay compared to when the RNA comes up. But nevertheless, for our purposes, it can be used to see what happens to the head organizer when the Hydra reverses its regeneration. So the fluorescent signal that you see here is the Win3 activity. What you see on the right is just a marker of the epithelium, fluorescence marker in another color, in red, just to show you along the way that the fluorescence did not go away. It's the Win3 actually that goes away. So let me run it quickly. We put a field above criticality, and you see the Hydra goes back. The fluorescent signals goes away. We can observe this for a long time to see that it's not because it rotated and etc. And then we drop the external voltage to 0, and the system is alive. You see that GFP starts to come back, and you see a very good signal of the system. And then you can go back and do cycles and see that the major component of the head organizer actually decays when the system goes back. And this is a summary of statistics over 7 tissues that shows you how it decays. It's a measure of the dynamics. And the error is very small. This is the shadow here, is a standard error. And this is a specific trajectory of one of the tissues that we could follow a long time, increasing the voltage and then dropping it to 0 and start to see the signal coming back. Again, I wouldn't trust the dynamics literally here, just the trend, because there is a gap between the fluorescence activity and the molecular biology that goes behind it. But if it does not exist, there is no activity. If it does exist, there must be already activity. Because it's autocatalytic, it maintains itself and amplifies itself to some level. Now, why reversal development is an exciting surprise? So again, historically, development, the term development was coined because people thought about development of a picture capture on a camera film. Really, literally, the word development came because people thought that animal develops like you develop a film. The pattern emerges because of chemicals that are doing something. Then they realize that it's not true. But then and the idea of epigenesis came in. I don't have time to elaborate on this. But this was extended to modern time by the concept of a program. There is a program, a hierarchical program that feed forward to the system. So this is the capture in also developing cell types, specific cell types for development. And then a surprise came, which is not so much a surprise, but a useful technique that was developed by Yamanaka and other people who got the Nobel Prize for it, that you can take a cell that is fully developed and perturb it back to become a stem cell. This is called reprogramming. And the question that is still open for us, is a fully developed animal also reprogrammable? Up to a level, of course, I don't expect to take a mouse, a fully developed mouse, and bring it back to be an egg or something, okay? It would be very useful to do it on people, by the way, if you can bring them back to some early stages in their development and develop different trajectories. I think they try to do it to Trump now, but unsuccessfully. Anyway, so it's a big question. This was mostly shown in vitro, and in the living system environment in development, the question how much of the processes are really reversible, it's a completely open question. So I'm already really beyond time. Let me just summarize. So what I showed you on electric fields, there exists a critical fields that halts the system. In this experiment that I briefly showed here, we can go to hundreds of hours, literally hundreds of hours. We can hold the sample and measure it for a very long time. And this is another thing that I learned about biology, is that if you open the measurement time, you start to see modes and phenomena that you didn't realize before because you look only on short timescales, only on transient phenomena. So opening timescales is really important. Here you can open, in this experiment, we move the voltage to study all kinds of things, but you can hold the tissue without losing regeneration potential and without actually regenerating for hundreds of hours. Let me go back to the summary. So the re-emerged body plane is not necessarily similar when you reinitiate trajectory. So this is very interesting because you can study the potential of the system to develop. Clearly, and this we showed in a recent publication, I think it's completely convincing, that reversal of morphogenesis is triggered by a non-electrical excitation, and it also leads to the decay of the head organizer. So it's not only morphology that goes back, and there is adaptation that you can see by different ways. So over time, the tissue adapts and critical voltage changes, all those the spatial temporal dynamics become sensitive to the history of the system. And for me, calcium activity is really crucial to understand because first, Hydra is a muscle, basically, and calcium plays a role in all muscles that we know. It's a muscle that, by the way, is not well understood. It's not part of the classical muscles that were studied for many years. But calcium activity is also a very good way of thinking on integrator of the different types of processes, biochemical, mechanical, and electrical. And finally, the implications are the fact that you can reverse the process and modulate it by external fields in a controlled way tells you that you have to go beyond biochemical patterning. That's obvious, but you have to go beyond the idea that development is just a forward-driven hierarchical process, so a program that it just runs subroutine by subroutine to create another organ until you get the final element. Clearly, electrical processes play an instructive role, so much so that since we can amplify or control their importance, we can tune it to the place where mechanics is more important, biochemistry is more important, and then to a level that electrical processes can become more important. And this is very important to understand this junction of the three fields that I mentioned at the beginning. Electrical processes are tightly integrated and coordinated, clearly with the biochemical and mechanical processes. The question whether developmental processes are fully reversible is open, completely open. We need to put more probes to look on more processes under electric field and start to understand how much of the system really goes back. And the last remark, and I'll finish by that, is that because developmental systems, any biological system, but developmental systems are historical systems, again, not from the perspective of evolution, but from the perspective of development itself, it takes a trajectory that steps along the trajectory, depends on what happened to the system along the trajectory. To understand the developmental potential of the system, rather than just one realization of it, using perturbation like the one I showed with the electric field, are absolutely essential to develop the methodology that we are lacking of how to deal and how to build eventually a physical theory for understanding the dynamics in historical systems. So this summarizes what I say, and again, the essence is closing the loop, and I think any perturbation that works on a junction, interesting junction between biochemistry, mechanical, and electrical processes is essential to understand how the system closes the loop. And finally, let me acknowledge, again, Kinneret mentioned this, we collaborate on hydromechanics and polarity, essentially. And Hillel Ori was a student here, helped with electrical measurements, and I think I should stop here. Thank you very much.
Thank you very much, Erez. That last part was quite stimulating. So there are, as one expects, many questions. So let's start. In the interest of time, I'm going to collate a few because there are a few themes that have come up in the questions. And some of the discussion may also move to Slack.
Would I leave it shared? Would I leave it shared or move it?
So, yeah, you can
okay.
Leave the slides right now, and then I'm going to invite people to ask you questions. Okay. Shanti, you want to ask a question?
Hi. Yeah, thank you for this really, really beautiful talk.
Okay, I guess I'll ask.
Can you hear me? Hello?
Yeah, go on.
I hear you, yes.
Hi, yeah. Thank you for this really beautiful talk. The idea that you can reverse development experimentally is, yeah, I don't know, I'm kind of speechless about it. I was wondering, there are tons of questions that come to mind, but you said that the organism maintains a memory, but every time you change the voltage, it doesn't necessarily return back to the same form. So for example, it doesn't always have the same number of tentacles. I was wondering if you've done this experiment with budding Hydra, so that, you know, maybe the wind signaling has already started and there's an additional head organizer to see if, you know, the bud river comes back as well. And then I also had a question about what's happening in that sort of stagnant stage where you said you can pause it for hundreds of hours. I was wondering if you could speculate on what's happening in that, you know, spheroid in those hundreds of hours. Yeah.
Thank you. These are excellent questions. So we didn't do experiments on buds, but if you take adult Hydra, you can reverse it. But not, I didn't do anything specific on buds, so we didn't do any experiments on this. The question about, so sorry, your second question, maybe I missed the, can you just?
Yeah, I was just asking what's happening in those, you said you could pause it for hundreds of hours. What's happening in those hundreds of hours? If you could speculate on that.
The short answer is I don't know. The longer answer that also starts by I don't know, but I think that what happens is there are two features of Hydra that really makes an electrical perturbation by external fields very useful. One I mentioned is the fact that the cells are excitable. The other is related, and I wonder if there are, you know, even more general aspect to this, is the fact that Hydra is a muscle, so it's a developing muscle in essence. And because it's a developing muscle, once you perturb the electrical processes, it really touches a junction that has to do with signaling mechanics and electrical processes. So what I think the electrical perturbation is doing is it perturbs the electrical communication between the system and slaves it to something that is driven by the external field instead of being driven by the internal self-organization dynamics. And by doing that, you stop the process at some stage. In fact, you can stop the process at different stages. I didn't talk about it. You don't have to take it back to a spheroid. You can really hold the system at the edge of regeneration. You see that it starts to get tentacles out and then it takes them back. It starts to take them back. And the reason I think is that you perturb the system in such a way that you put stresses, both electrical stresses and mechanical stresses, into the system that the system needs to accommodate by halting the normal trajectory. But these are, at the moment, we don't know more than this. One of the issues that I would like very much to go into is to see, for example, how actin organization, we didn't do serious work on this yet, how actin organization is affected by the electric field. What we could show is that actin orientation is not affected by the external field. So the actin itself does not behave as a polar electrical agent. But however, and this we have long-term arguments, is the question whether we can get Hydra to live with actin at the isotopic phase rather than the nematic phase. I'm not talking about regeneration, I'm talking about keeping the regeneration potential and melt the actin order by the external electric field. So these are different trajectories that we now start to take in the research. So if this could be done, this is another way also to perturb an important mechanical process, bring it to this order and see how order is rebuilt. At the moment, I don't know more than what I just said.
Thank you, Erez. In the interest of time, I'm going to, I guess, in your answer just now, you answered, by the way, a couple of more questions. For example, Charles's question on the annihilation of topological defects. But there are a few questions which I'm going to condense relating to calcium activity. So does the calcium activity do anything to the ion channels? And do you think some of that is responsible for this reversal or stasis behavior that you see? And also, I guess a related question then is, does that naturally relate to some kind of a threshold voltage beyond which some of these effects go away?
Calcium ion channels must play important role. I tried to block certain channels. I didn't see much effect, but one has to understand there is a zoo of channels. Most of them in Hydra are not well explored. And the question is how to block and perturb ion channels is not a simple thing, because you can, there are many ways to kill the system. And if you really block in an aggressive way some essential process, you can easily kill the system. So this is part of what we do now, is to try all kinds of blockers, not only for channels, also for gap junctions, to see if we can modulate the electrical effect across the system, and by this trying to understand what the electric field is really doing. So it's related also to the previous question. One way to go is to perturb intrinsic internal electrical processes and see how they affect the process in order to understand what exactly the external field is doing by these excitations to the tissue.
Thank you. So there are also some general questions regarding complex systems and self-organization, etc. So I guess we can move some of those discussions onto Slack in the interest of time here. So I guess unless there are any other announcements from the organizers, let's thank Kinneret and Erez for these very nice talks on the physics of morphogenesis. I'm sure they will stimulate a lot of ideas and discussion, and I hope a lot of you will take that to the Slack channels and even beyond. So thanks very much, Erez. That was, if I may say, electrically stimulating talk.
Thank you, Erez. Thank you.